Today we are talking about how cellular energy is created from eating Dr. Spencer's world-famous cupcakes. Although we don't have a nutrition facts label for these cupcakes, let's assume that it has approximately the same nutritional value as an average cupcake, which means one serving would contain about 17 grams of carbohydrates. As you know, the body breaks down carbohydrates into simple sugars, like glucose, which your cells can then use to create energy. This energy comes in the form of adenosine triphosphate, or ATP, the energy currency of your cell, and the process by which your cells create this energy is called cellular respiration. There are 3 stages to cellular respiration: glycolysis, the Krebs cycle, and the electron transport chain. The first stage is called glycolysis, which happens in the cytoplasm, and is a process that does not require oxygen but does require some energy. So, first, an enzyme called hexokinase, with the help of ATP, catalyzes the phosphorylation of glucose to produce 2-glucose-6-phosphate, which is a more reactive form of glucose. Second, phosphoglucose isomerase converts glucose 6-phosphate into fructose 6-phosphate, so that'll be easier to split later. Third, the enzyme phosphofructokinase, with the help of another molecule of ATP, catalyzes the phosphorylation of fructose 6-phosphate into fructose 1,6-biphosphate. Fourth, an enzyme called aldolase splits fructose 1,6-biphosphate into 2 3-carbon isomers, which are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Fifth, those triose phosphate isomerase transforms the dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. So now we have 2 molecules of glyceraldehyde 3-phosphate. In step 6, the glyceraldehyde 3-phosphates are oxidized to extract high-energy electrons, which are then carried away by NAD to produce NADH. At the same time, the glyceraldehyde 3-phosphate is phosphorylated again, producing 1,3-biphosphoglycerate. In step 7, an enzyme called phosphoglycerate kinase forces the 1,3-biphosphoglycerate to donate a phosphate group to ADP, forming 1 molecule of ATP. The 1,3-biphosphoglycerate is oxidized to form 3-phosphoglycerate, and in step 8, phosphoglycerate mutase catalyzes the movement of the 3rd carbon to the 2nd carbon, turning the 3-phosphoglycerate into 2-phosphoglycerate. In step 9, enolase causes 2-phosphoglycerate to undergo a dehydration reaction to produce phosphoenolpyruvate, and in the 10th and final step of glycolysis, the enzyme pyruvate kinase catalyzes the phosphorylation of phosphoenolpyruvate, resulting in the production of pyruvate and ATP. So the total end products of glycolysis are 4 ATP molecules, 2 NADH molecules, and 2 pyruvate molecules. In order to make use of pyruvate, it needs to be transformed. So in the mitochondrial matrix, a carboxyl group is removed from pyruvate, which produces carbon dioxide as a waste product, and NAD is reduced to NADH, and an acetyl group is transferred to coenzyme A, resulting in a molecule of acetyl-CoA. Now the Krebs cycle, or citric acid cycle, which occurs in the mitochondrial matrix, starts with the condensation of the acetyl-CoA and oxaloacetate to form citrate. Next, citrate is converted to isocitrate by a conitase. Third, with isocitrate dehydrogenase, the isocitrate is converted to produce alpha-ketoglutarate, carbon dioxide, and 2 electrons, which reduces NAD to NADH. Fourth, the alpha-ketoglutarate dehydrogenase catalyzes alpha-ketoglutarate to be converted to succinyl-CoA. More carbon dioxide is released, and a molecule of NAD is reduced to NADH. In step 5, succinyl-CoA is turned into succinate by succinyl-CoA synthase, generating a molecule of GTP. Sixth, succinate is converted to fumarate with succinate dehydrogenase. Seventh, fumarate is turned into malate by the enzyme fumarase. And finally, eighth, malate dehydrogenase turned malate into oxaloacetate, and a molecule of NAD is reduced to NADH. This completes the cycle, and oxaloacetate can be converted to citrate using citrate synthase to start the cycle all over again with additional acetyl-CoA. The total end products of the Krebs cycle from one glucose molecule is 6 NADH molecules, 2 FADH2 molecules, 2 GDP or ATP molecules, and 4 CO2 molecules. The NADH and FADH molecules will connect with the last stage to produce ATP molecules. The last stage of cellular respiration is the electron transport chain, which occurs inside the mitochondria between the inner membrane and the intermembrane space. Here, a series of redox reactions transfers electrons between electron donors and electron acceptors. The flow of electrons is an exergonic process, which creates a proton gradient that powers the synthesis of ATP, similar to how water might power a turbine. So, 4 protein complexes embedded in the inner membrane help maintain this gradient. Complexes 1, 3, and 4 pump protons into the intermembrane space, while complex 2 does not. NADH carries 2 electrons to complex 1. The electrons are passed along a chain of redox centers, which creates energy. Complex 1 uses this energy to pump protons across the membrane, establishing the gradient. Then coenzyme Q takes 2 electrons from complex 1 and brings it to complex 3. Meanwhile, complex 2 directly receives electrons from FADH and does a similar thing to complex 1. It lets the electrons run through the redox centers before donating them to coenzyme Q. However, complex 2 does not use the harnessed energy for proton pumping. Now coenzyme Q brings the donated electrons to complex 3, where 1 electron passes through 2 redox centers before being carted off to complex 4 by cytochrome C. And in complex 4, electrons convert a molecule of oxygen to water, further strengthening the gradient. And these 4 complexes together maintain the proton gradient that enables the large protein complex F1F0 ATP synthase to keep synthesizing ATP molecules.